Hello students, our today's topic is going to be incomplete and codominance, multiple alleles, lethal alleles, epistasis and pleiotropy. Incomplete dominance. Incomplete dominance, also called partial dominance or semi-dominance, occurs when the phenotype of the heterozygous phenotype is distinct from and often intermediate to the phenotypes of the homozygous phenotypes. For example, the snapdragon flower color is homozygous for either red or white. When the red homozygous flower is paired with the white homozygous flower, the result yields a pink snapdragon flower. The pink snapdragon is the result of incomplete dominance. A similar type of incomplete dominance is found in the 4-o'clock plant, wherein pink color is produced when true bred parents of white and red flowers are crossed. In quantitative genetics, where phenotypes are measured and treated numerically, if a heterozygote's phenotype is exactly between that of the two homozygotes, the phenotype is said to exhibit no dominance at all. That is, dominance exists only when the heterozygote's phenotype measure lies closer to one homozygote than the other. Codominance. Codominance occurs when the contributions of both alleles are visible in the phenotype. For example, in the ABO blood group system, chemical modifications to a glycoprotein, the H antigen on the surface of blood cells, are controlled by 3 alleles, 2 of which are codominant to each other and dominant over the recessive allele at the ABO locus. The allele A and B alleles produce different modifications. The enzyme coded for by allele A adds an N-acetyl galactosamine to the membrane-bound H antigen. The allele B enzyme adds a galactose. The recessive allele produces no modification. Thus, allele A and allele B are each dominant to recessive. Individuals both have type A blood and dominant B alleles, and allele B dominant and recessive individuals both have B blood. But allele A and allele B individuals have both modifications on their blood cells and thus have type AB blood. So, the allele A and allele B are said to be codominant. Codominance, where allelic products coexist in the phenotype, is different from incomplete dominance, where the quantitative interaction of allele products produces an intermediate phenotype. For example, in codominance, a red homozygous flower and a white homozygous flower will produce offspring that have red and white spots. When plants of the F1 generation are self-pollinated, the phenotypic and genotypic ratio of the F2 generation will be 1:2:1. These ratios are the same as those for incomplete dominance. Multiple alleles. The word allele is a general term to denote the alternative forms of a gene or contrasting gene pair that denote the alternative form of a gene is called allele. These alleles were previously considered by Bateson as hypothetical partner in Mendelian segregation. In Mendelian inheritance, a given locus of chromosome was occupied by 2 kinds of genes, that is, a normal gene for round seed shape and other its mutant recessive gene, a wrinkled seed shape. But it may be possible that normal gene may show still many mutations in P besides the one for wrinkledness. Here the locus will be occupied by normal allele and its 2 or more mutant genes. Thus, 3 or more kinds of genes occupying the same locus in individual chromosome are referred to as multiple alleles. In short, many alleles of a single gene are called multiple alleles. The concept of multiple alleles is described under the term multiple allelism. Characteristics of multiple alleles. The study of multiple alleles may be done in population. Multiple alleles are situated on homologous chromosomes at the same locus. there is no crossing over between the members of multiple alleles. Crossing over takes place between two different genes only intergeneric recombination and does not occur within a gene, intragenic recombination. Multiple alleles influence one or the same character only. Multiple alleles never show complementation with each other. By complementation test, the allelic and non-allelic genes may be differentiated well. The production of a wild-type phenotype in a trans-heterozygote for two mutant alleles is known as complementation test. The wild-type normal allele is nearly always dominant, while the other mutant alleles in the series may show dominance or there may be an intermediate phenotypic effect. When any two of the multiple alleles are crossed, the phenotype is of a mutant type and not the wild type. Further, F2 generations from such crosses show typical monohybrid ratio for the concerned character. Examples of multiple alleles. Wings of Drosophila. In Drosophila, wings are normally long. There occur two mutations at the same locus in different flies, one causing vestigial wings and the other mutation causing antillared, less developed wings. Both vestigial and antillared are alleles of the same normal gene and also of each other and are recessive to the normal gene. Lethal alleles. Lethal alleles are alleles that cause the death of the organism that carry them. They are usually a result of mutations in genes that are essential to growth or development. Lethal alleles may be recessive, dominant, or conditional depending on the gene or genes involved. Lethal alleles can cause death of an organism prenatally or any time after birth, though they commonly manifest early in development. Types of lethal alleles. Recessive lethal. A pair of identical alleles that are both present in an organism that ultimately results in the death of that organism are referred to as recessive lethal alleles. Though recessive lethals may encode for dominant or recessive traits, they are only fatal in the homozygous condition. Heterozygotes will sometimes display a form of disease phenotype as in case of achondroplasia. One mutant lethal allele is tolerated but having two results in death. In the case of homozygous achondroplasia, death almost invariably occurs before birth or in the prenatal period. Not all heterozygotes for recessive lethal alleles will show a mutant phenotype, as is the case for cystic fibrosis carriers. If two cystic fibrosis carriers have children, they have a 25% chance of producing offspring having two copies of the lethal allele, eventually resulting in the death of the child. Another example of a recessive lethal allele occurs in the max cat. Max cat possesses a heterozygous mutation resulting in a shortened or missing tail. Crosses of two heterozygous max cats results in two offspring displaying the heterozygous shortened tail phenotype and one offspring of normal tail length that is homozygous for a normal allele. Homozygous offspring for the mutant allele cannot survive birth and are therefore not seen in these crosses. Dominant lethals. Alleles that need only be present in one copy in an organism to be fatal are referred to as dominant lethal alleles. These alleles are not commonly found in populations because they usually result in the death of an organism before it can transmit its lethal allele onto its offspring. An example in humans of a dominant lethal allele is Huntington's disease, a rare neurodegenerative disorder that ultimately results in death. A person exhibits Huntington's disease when they carry a single copy of a repeat expanded Huntington allele on a chromosome 4. Conditional lethals. Alleles that will only be fatal in response to some environmental factor are referred to as conditional lethals. One example of a conditional lethal is favism, a sex-linked inherited condition that causes the carrier to develop hemolytic anemia when they eat fava beans. Epistasis. Epistasis is the phenomenon of the effect of one gene being dependent on the presence of one or more modifier genes. Originally, the term meant that phenotypic effect of one gene is masked by a different gene locus. Thus, epistatic mutations have different effects in combination than individually. It was originally a concept from genetics, but it is now used in biochemistry, computational biology, and evolutionary biology. It arises due to interactions either between genes or within them, leading to nonlinear effects. Epistasis has a large influence on the shape of evolutionary landscapes, which leads to profound consequences for evolution and evoluability of phenotypic traits. Classification. Epistasis is used to denote the departure from independence of the effects of the different genetic loci. Confusion often arises due to the varied interpretation of independence among different branches of biology. The classifications below attempt to cover the various terms and how they relate to one another. Additivity. Two mutations are considered to be purely additive if the effect of the double mutation is the sum of the effects of the single mutations. This occurs when genes don't interact with each other. For example, by acting through different metabolic pathways. Simple additive traits were studied early on in the history of genetics, however, they are relatively rare, with most genes exhibiting at least some level of epistatic interaction. Magnitude epistasis. When the double mutation has a fitter phenotype than expected from the effects of two single mutations, it is referred to as positive epistasis. Positive epistasis between beneficial mutations generates greater improvements in function than expected. Positive epistasis between deleterious mutations protects against the negative effects to cause a less severe fitness drop. Conversely, when two mutations together lead to a less fit phenotype than expected from their effects when alone, it is called negative epistasis. Negative epistasis between beneficial mutations causes smaller than expected fitness improvements, whereas negative epistasis between deleterious mutations causes greater than additive fitness drops. Independently, when the effect on fitness of two mutations is more radical than expected from their effects when alone, it is referred to as synergistic epistasis. The opposite situation, when the fitness difference of one double mutant from the wild type is smaller than expected from the effects of the two single mutations, it is called antagonistic epistasis. Therefore, for deleterious mutations, negative epistasis is also synergistic, while positive epistasis is antagonistic. Conversely, for advantageous mutations, positive epistasis is synergistic, while negative epistasis is antagonistic. The term genetic enhancement is sometimes used when a double deleterious mutant has a more severe phenotype than the additive effects of the single mutants. Strong positive epistasis is sometimes referred to by creationists as irreducible complexity, although most examples are misidentified. Sign epistasis. Sign epistasis occurs when one mutation has the opposite effect when in the presence of another mutation. This occurs when a mutation that is deleterious on its own can enhance the effect of a particular beneficial mutation. For example, a large and complex brain is a waste of energy without a range of sense organs. However, sense organs can be more useful if the organism's brain is better able to process the information. At its most extreme, reciprocal sign epistasis occurs when two deleterious genes are beneficial when together. For example, producing a toxin alone can kill a bacterium, and producing a toxin exporter alone can waste energy. But Producing both can improve fitness by killing competing organisms. Reciprocal sign epistasis also leads to genetic suppression, whereby two deleterious mutations are less harmful together than either one on its own. That's one compensates for the other. This term can also apply sign epistasis, where the double mutant has a phenotype intermediate between those of the single mutants, in which case the more severe single mutant phenotype is suppressed by the other mutation or genetic condition. Pleiotropy. Pleiotropy occurs when one gene influences two or more seemingly unrelated phenotypic traits. Therefore, a mutation in a pleiotropic gene may have an effect on several traits simultaneously due to the gene coding for a product used by a myriad of cells or different targets that have the same signaling function. There are three types of pleiotropy: molecular gene pleiotropy, developmental pleiotropy, and selectional pleiotropy. The different types are used to help categorize the different types of pleiotropy that can occur, but they are not exclusive to one another since the different types do overlap. Molecular gene pleiotropy is seeing how many traits are affected by a molecular gene. Developmental pleiotropy is a mutational pleiotropy observing the relationships between mutations and different phenotypes, excluding fitness. Selectional pleiotropy is evaluating how a mutation affects fitness. An example of pleiotropy is phenylketonuria, which is an inherited disorder that affects the level of phenylalanine in the body. Phenylalanine is an amino acid that can be obtained from food. Phenylketonuria causes this amino acid to increase in amount in the body, which can be very dangerous. The human disease is caused by a defect in the single gene on the chromosome 12 that affects multiple systems such as the nervous and integumentary system. Other examples of pleiotropy are albinism, sickle cell anemia, and certain forms of autism and schizophrenia. Pleiotropy not only affects humans but also animals, such as chickens and laboratory house mice, where the laboratory house mice have found to exhibit the mini-muscle allele. Pleiotropic gene action can limit the rate of multivariate evolution when natural selection, sexual selection, or artificial selection on one trait favors one specific version of the gene, while selection on other traits favors a different allele, which shows how evolution is negatively related with pleiotropy in models. Although some of the evolution in genes can be beneficial, some gene evolution is harmful to an organism. Genetic correlations and responses to selection most often exemplify pleiotropy. Examples of pleiotropy: albinism. Albinism is the mutation of the tyrosine gene, also termed tyrosinase. This mutation causes the most common form of albinism. The mutation alters the production of melanin, and which affects traits all throughout the organism. Melanin is a substance made by the body that is used to absorb light and provides coloration to the skin. Indications of albinism are the absence of color in an organism's eyes, hair, and skin due to the lack of melanin. Some forms of albinism are also known to have symptoms that manifest themselves through rapid eye movement, light sensitive, and strabismus. Phenylketonuria, a common example of pleiotropy, is the human disease phenylketonuria. This disease causes mental retardation and reduced hair and skin pigmentation, and can be caused by any of a large number of mutations in the single gene on chromosome 12 that codes for the enzyme phenylalanine hydroxylase, which converts the amino acid phenylalanine to tyrosine. Depending on the mutation involved, conversion of phenylalanine to tyrosine is reduced or ceases entirely. Unconverted phenylalanine builds up in the bloodstream and can lead to levels that are toxic to the developing nervous system of newborn and infant children. The most dangerous form of this is called classic phenylketonuria, which is common in infants. The baby seems normal at first but actually incurs permanent intellectual disability. This can cause symptoms such as mental retardation, abnormal gait and posture, and delayed growth. Because tyrosine is used by the body to make melanin, a component of the pigment found in the hair and skin. Failure to convert normal levels of phenylalanine to tyrosine can lead to fair hair and skin. The frequency of this disease varies greatly. Specifically, in the United States, phenylketonuria is found nearly 1 in 10,000 births. Due to newborn screening, doctors are able to detect phenylketonuria in a baby sooner. This allows them to start treatment early, preventing the baby from suffering from the severe effects of phenylketonuria. Phenylketonuria is caused by a mutation in the phenyl hydroxylase gene. The role of the phenylalanine hydroxylase gene is to instruct the body on how to make phenylalanine hydroxylase. Phenylalanine hydroxylase is what converts the phenylalanine taken in through diet in other things that the body can use. The mutation often decreases the effectiveness or rate at which the hydroxylase breaks down the phenylalanine. This is what causes the phenylalanine to build up in the body. The way to treat phenylketonuria is to manage one's diet. Phenylalanine is ingested through food, so a diet should decrease types of foods that have high contents of phenylalanine. Foods with high levels of protein must be avoided. These include breast milk, eggs, chicken, beef, pork, fish, nuts, and other foods. A special phenylketonuria formula can be obtained in order for the body to have protein. That's all for today's lecture. Thank you very much.